Started. Today we have the pleasure of having Angus Forbes here from the University of Illinois at Chicago. He's an assistant professor of computer science and part of the electronic visualization lab. He's here interviewing for one of the Transformative Excellence Program clusters in biodiversity. So we have the Biodiversity Search Committee here as well. Welcome to the Ski Institute. And there's computer science faculty, and there's Ski Institute faculty, and staff and students. So welcome, everybody. So I only found out yesterday that after Angus received his bachelor's degree in literature, that 20 years ago, he lived on 3rd Avenue and was a PhD student in English here for a couple of years. And then he got bit by the technology and media bug, I would say, and went on to get master's and PhD from UC Santa Barbara in media arts and computing. Spent a couple of years in Arizona, and then for the last 4 years, he's been at the University of Illinois at Chicago. And he's done some really interesting work in information visualization for large data and scientific applications, and also on the creative side of using computing and computers and visualization for creative art. And I think he'll talk a little bit about both of them and maybe more on the information visualization side on this talk. So please join me in welcoming Angus Forbes.
Thanks, Chris, for the introduction. Can you hear me? Sounds like you can. So as Chris mentioned, I'm currently at University of Illinois at Chicago, where I'm a research faculty at the Electronic Visualization Laboratory and where I direct the Creative Coding Research Group. In addition to being an assistant professor in the computer science department, I have an affiliation with the College of Architecture, Design, and the Arts, and I teach courses in computer graphics, visualization, visual analytics. I teach a course called Creative Coding. I introduce a course called Computational Aesthetics, and most recently I've been teaching a course on deep learning. So I gave a presentation yesterday at the Natural History Museum where I talked a little bit more about some of the arts installations and creative installations and public artworks that I've developed, most of which do involve data in some capacity. And today I'm going to talk more about various information visualization projects. And so these are just some of the students and postdocs I work with or have worked with in the last few years. And this is a more recent picture of some of the people in my lab, undergraduates, graduate students, and postdocs. So some of the work you'll see today is our collaborations with them. So I just want to, I'll try and name them, but if I don't, that's what they look like. So some of my research interests recently, I guess I should start off by saying, when I was studying at UC Santa Barbara, when I was studying media arts, I was very interested in personal expression and kind of my personal response to cultural archives and data sets. And more recently, I've been interested in working with scientific data sets, partly because I think in one sense, they're just more interesting. They're more complicated. No one really understands how best to represent certain types of data. So it's an interesting place to think both scientifically and empirically, but also very creatively. And so I've been doing a lot of work in the last few years on promoting a visualization symposium, an art symposium within IEEE Viz, the main visualization conference, that brings together designers and artists to think about representation, interaction, and different ways of conceiving data. So, but nonetheless, one of my main interests is thinking about how to represent, interact with, and reason about dynamic big data to solve interesting, difficult, real-world problems, and how to use visualization to understand complex systems and the interaction within and between systems. So in addition to trying to represent complicated data, that data is part of a more complicated system, and the whole point of representing data oftentimes is to model these systems. And I think an exciting area of data visualization is not only modeling the systems, but modeling how those systems interact with each other, maybe at different scales. And this is kind of one of the main points I want to get across in my transformation from thinking about subjective expression to data visualization, is that I think data visualization can be translational in a clinical sense, and that I want data to not just be about representation, but about action. I want to move from observation to action and potentially from diagnosis to an intervention in the same way a doctor might or a nurse might. So today I'm going to talk about some BioVis projects, but before I do, I just want to also mention that I'm very interested in this idea of immersive analytics, or using VR as a way to become more engaged with data visualization. And just briefly, this is a project I did in collaboration with Bashak Alper when we were both at University of California, Santa Barbara, where we're using the Alisphere to do some research into how well people could perceive node link diagrams in 3D, especially in VR, using stereoscopic glasses. And I bring this up because, as maybe some people here know, there's a little bit of a stigma, I would say, about using 3D displays or using VR for information visualization. It's seen as kind of gimmicky or maybe not useful. And I think that comes out of perhaps the 80s and the 90s during one of the first waves when VR was popular, but it wasn't particularly high enough resolution. And so it wasn't actually effective. It wasn't perceptually an effective way to present data. So now that we have much better display technologies, including portable display technologies, there's been a reinvestigation into the use of VR for data visualization. And so this is a project from, I think, 5 or 6 years ago now, or maybe even 7 years ago, where we wanted to see what the differences were between presenting a complicated node link diagram in 2D with various visual encodings versus in 3D. And it turns out they're about equivalent in some cases, but 3D is actually better in some situations. If you can, for example, I'm looking right now at an audience where people are in rows, and the human brain is able to see things on different planes pretty easily. Whereas if you were grouped, kind of clumped together, it would be harder to perceive links and do kind of basic tasks and graph reading. So that was kind of an interesting result. Now that I'm at UIC, we have the Cave 2, which is, I'm sure most of you are familiar with the Cave, which is usually back projected, 6-sided or 4-sided sometimes, square that you can go into and wear goggles. This is the new version created by Jason Lee and Andy Johnson, some of the other researchers who still are at UIC, using high-resolution 5K displays or 72 displays, so you get very dense pixel resolution, and you can actually display very detailed data. Again, I'm talking more about BioVis today, but I've also been working in collaboration with some of the neuroscientists and clinical neuroscientists at UIC to represent their data, both in the Cave and more recently on portable devices. The problem with these big room immersive VR displays, of course, is you have to go to them. They're not portable, so you have to book time in them. They're also quite expensive to run. And for some tasks, just using off-the-shelf Oculus Rifts or HTC Vives, or in some case even the Google Cardboards, is sufficient to do certain data analysis tasks. So we've been researching ways to represent MRI data in VR and to facilitate various comparison tasks in VR and to apply different dimensionality reduction techniques to representations of brain voxels or neuron activity, and to be able to do things like simulate lesions to see what the human brain connectome would look like in different topological spaces, and also to represent different types of functionality in response to tasks that were recorded with MRI machines. So, as I mentioned, my current interests are about how to represent, interact with, and reason about complex dynamic data. I just showed you some slides working with the human connectome data, various human connectome data sets, to think about how we can measure and potentially affect how we think about and respond to the world. I've been working on some health informatics projects, thinking about how we can get the right information to the right people at the right time in clinical environments and hospitals, and especially working with nurses to see if we can facilitate better communication between nurses when they switch. They create what are called shift summarization reports, and oftentimes they don't actually hand them off to each other or talk to the other nurses. They just enter it into an EHR or EMR, and then the nurse has to read it. And I don't know if you've had a chance to look at EHRs, but in general, they're not the most well-designed interfaces. So seeing if we can overlay a different type of a better designed visualization on top of those interfaces to facilitate communication is an ongoing project funded by National Institutes of the Health. And particularly, it's a smart and connected health project. I'm also interested in cultural data, what the most compelling ways are to represent or to highlight meaningful relationships between data, text, and image. And as I'm going to talk about more, biological pathway data to try and understand what happens inside a single cell and to figure out how we can best represent our own biochemistry. So something I've been thinking about a lot is this idea of representation, especially in thinking about cells. So my initial instinct when given interesting systems biology data, molecular biology data about the cell, was to try and do kind of a realistic visualization or an approximation of a realistic visualization of what happens inside a cell. But there's so much going, it's so complicated, there's so much going on, it's actually not really possible to do that. And so representation, photorealistic or a facsimile of photorealistic simulation is actually not enough, and in fact hinders sometimes. And I ask the question, so what if we can see it if we don't understand it? What insight does the data give us about our world, about the different systems the data is sampled from and that we are connected with? And a second kind of overarching question is this comment that accurate, useful interpretation of data is often much more important than the raw data itself. We need to figure out, visualization should be used to figure out what to do with the data and to figure out what the data means and in what context the data has meaning. And lastly, data transparency and effective data communication is necessary in visualizations and sometimes is ignored in visualization projects. The tools to filter, clean, interpret, or transform raw data may introduce bias and obscure or distort meanings. Critical thinking is informed by understanding how humans, and more often how machines, are modeling the world, especially with the rise of the machine, with the popularity of machine learning algorithms and automated decision-making algorithms. So I've been thinking a lot about how we can best create visual design and visual analytics tools that provide information about the context of the data, that indicate the provenance of the data, where it came from and how trustworthy it is, that enables users to explore and to update the underlying models of the data to facilitate new interpretation, to think about different visual encodings that can represent ambiguity or depict probabilistic functionality more effectively, and that take advantage of the innate human ability to make sense of the world through storytelling. And finally, to allow users to interactively test hypotheses by simulating data. So all the projects I'm going to talk about over the next 20 or 30 minutes are funded, at least in part, by the Big Mechanism Project. And this is a multi-year DARPA project that I think ends at the end of 2018. And it's an interdisciplinary collaboration involving a team with expertise in molecular biology, systems biology, bioinformatics, computational linguistics, machine learning, and visualization. And it's an interesting project. The overall goals of the project are, one, to better understand intercellular mechanisms of how cells work, and especially how they might go wrong and lead to tumor growth, and potentially how to stop that tumor growth. But number two, more broadly speaking, to figure out how to develop computational systems that can better utilize knowledge that already exists. And this always wows me. Cancer biology, what happens inside a human cell is insanely complicated. We have up to 1 million proteins in a single cell in our body, and we have over 37 trillion cells in the human body. So trying to make sense of the functionality of living systems is a kind of unimaginably complicated. So the goal of the Big Mechanism Project is kind of a natural linguistics processing project to read pathway information, to extract knowledge from PubMed articles, and specifically to extract what they're calling causal fragments, and potentially to assemble these new to assemble new pathway networks from these articles, and then finally to use that knowledge to explain causes and effects within biological systems and let domain experts answer various questions about the pathways. So the issue here is that being that human biology and cell biology is so complicated and protein analysis is so complicated, all the different labs, all the cancer research labs, even if they're all working as hard as they can, are only scratching the surface of what's going on. So the idea is if we can get, and moreover, any one person cannot possibly make sense of the complexity, or even one lab cannot make sense of the complexity of what's happening. So the idea is that if a machine could read all this information, at least in approaching the intelligence of a human, or even to some degree, then potentially it could fill in the blanks between what we don't know about a particular biological pathway, say. So the point there is that cancer biology knowledge is very fragmented, and it's also enormous, voluminous. And as I'll talk about in one of the projects, the knowledge is sometimes inconsistent, right? Different labs come to different results when they're looking at cell lines under different contexts. I like to put these statistics down sometimes because just to show how much knowledge is being created about something as simple as a human cell. There's 42,000 papers on cancer biology indexed in PubMed so far in 2017, and it's only May. As of April 10th, yesterday, even looking for papers that are about a particular protein that you might be interested in studying. There was 5,000 papers about the EGFR protein or the protein complex rather, in 2016, and RAS, which is indicated in certain cancers, 4,200 papers, individual papers about this one protein. So the NLP group, the big mechanism, the computational linguistics group that's doing the NLP for big mechanism at University of Arizona has created a system to automatically read, now it's up to 370,000 papers, academic papers about cancer biology that are open access on PubMed. That includes some information about biological pathways or the proteins or protein complexes indicated in biological pathways related to cancer signaling from 1977 all the way to today. So there's no way anyone could read that many papers. And they've extracted what they're calling knowledge fragments or causal fragments from these papers, around 10 or 40 pieces of information that relate to the mechanisms of how biology, of how biochemical reactions function in a cell. So the role of visualization here is to facilitate the answering of questions that are hard to answer. How can we illustrate relevant details regarding what happens inside a single cell? How can we best represent our biochemistry to make sense of some difficult biochemical problems? And how can we aid domain experts, which include systems biologists, molecular biologists, drug designers, oncologists, et cetera, in understanding and using their data and in reading, synthesizing, and potentially executing models of their papers. So I know some people here do bioviz, so you're probably familiar with diagrams like this. And this is a manual layout of a protein network. So again, researchers don't necessarily want to see the complexity. They want to see a summary or an overview related to a specific task or a specific area that they're working on. So they develop these manual layouts, which quite honestly I find difficult to read. And this is a very small pathway. Larger ones become more difficult to read, even when they're laid out manually. And the automated systems to try and do this, automate how a user might put together a pathway of interest, even in small networks, get quite complicated. And in larger networks become completely unreadable. So this is obviously a downfall of using node-link diagrams to represent dense networks, especially when there's issues with labeling, especially when the protein names are very long, especially when the protein names might be called different things by different people. So I was inspired by some of these beautiful poster-sized diagrams that you may have seen that were generated by Gerhard Michel, who's at Roche, but he developed them in the 60s, but he's still working on them. And these are enormous PDFs that try and summarize known knowledge about signaling pathways. So that's a kind of a close-up view. This is still just 1/50th of this whole poster. They're kind of nice looking. They look like these subway maps. They're the person who designed them, Gerard Michel, has an eye for design. But even though they're interesting, especially the older versions I find really pretty, they're static. There's no way to add new knowledge to them. Although you can kind of zoom in and zoom out, you can't really toggle things on and off. You can't look at only the data you care about. So they're not particularly useful. They're good for education, and maybe they're good for summarization, but they're not necessarily good for actual visual analysis tasks. So there's been different efforts to still utilize node-link diagrams or network layouts to try and come up with something that's interactive and useful, but they're still a mess. So this is Shibe, which came out of Memorial Sloan Kettering Cancer Center a couple years ago, and it's still an active project and still has many issues when you look at pathways that are larger than 50 or 60 nodes. So their goal here is to try and reduce edge crossing by introducing what they call node duplication. So you could have two of the same protein in different parts, and to try and mitigate some of the edge crossing, but then it just explodes and gets kind of difficult to read. So the goal is not, it is to come up with better representations, but not just to come up with better representations, but also to enable tasks that are useful to whatever the scientific audience is that's using it. So the kind of traditional one in network layouts is to tame the hairball. Everyone's seen networks like this that are, or especially like especially like this, where it's just a hairball of, or a bowl of spaghetti of connections. And to show the call, in this, particularly for this, the biologists we worked with, they were interested in seeing the causal relations within and between pathways. They wanted to see that things were connected, but they wanted to imagine what would happen if they were, for example, able to knock out a certain protein. How would that affect things downstream? And that could be useful potentially for drug design. If there was a drug that seemed to decrease the operations of one pathway, that might slow tumor growth. And to enable a wide range of analysis tasks, which I'll mention briefly for different domain experts. So, in sum, the biologists may have a clear sense of what components are in the cell, although actually they really don't, and how much of these components there are, but they want to know how they work, how energy flows between them, how information flows between them, when the biochemical actions occur, what they occur in response to, what order they happen in. And so one of the first projects that me and my colleagues at UIC worked on was just trying to, it's a little bit hard to see for some reason in this image. Oh, I don't think it's your projector, I think it's actually my slide. Is just trying to mitigate the complexity of some of these node link diagrams. And so one approach is to not use a node link diagram or to rethink how to use a node link diagram. So what we did is we kind of created this columnar layout where on either side we have the same set of proteins. And in the center of them are all the possible biochemical reactions that can occur with multiple proteins either being input or being transformed into something. And so what we were able to do is we created this animated arc diagram that flows through. If you start from any one place, or either one reaction or one or more sets of proteins, it animates through the network and shows a flow of what happens. And so this lets you do things like what happens if you knock out a protein, what's affected by it downstream. If you want to see, in this case, we're starting with 3 proteins to see what reactions are involved and then seeing what happens there. It's still a bit of a hairball, but because it's evolving over time and emerging over time, you get a better sense of how energy or how information flows and how these reactions flow in an order. And one of the things, if you think about tumor growth, what happens in cells is there's some disruption to the normal organization of the cell or the normal functionality of the cell, where you get these feedback loops where information or where proteins get in these feedback loops where they grow and they grow and they grow and they don't die. So identifying feedback loops or potential feedback loops in cellular systems is important. Given that, these networks are these systems are basically huge networks. Oftentimes, the biologist would want to find the shortest path between starting protein and end protein. What are all the possible reactions that could go from one protein or protein complex to another, and also to find common downstream components from any selected proteins or complexes. Kind of skim through these. So that's all that work assumes that the descriptions of the pathways make sense. But in fact, biologists are discovering new information about these pathways all the time. And that information is not stored in any one place. It's stored in different databases, sometimes in different file formats. And oftentimes some of the storage of the data actually overlaps. So one thing the biologists wanted to be able to do is to load up different files and see what was inside them. Was there overlap between proteins? Were some of the same pathways described, potentially even with different labels, in multiple file formats? So this is kind of a first stab at just taking that arc diagram visualization, reaction flow, and trying to see if we could have it work with multiple files simultaneously. Another visualization we came up with is called cactus trees. I'll just show this briefly, but the idea is to kind of almost think of this as like a file browser, right? The root node is a base pathway, and these base pathways may actually be hierarchical in structure and contain other pathways inside them, or have pointers to other databases. So this is kind of a quick way to summarize the complexity of multiple pathways interacting, starting from one point. So a user can kind of zoom into any part they want. They can open up certain side trees. They can turn certain ones on and off. And then what we were trying to do is think about how we could show the process, the causality of these biochemical reactions happening over these different pathways. And that just kind of as a tangent, this also led us to think about hierarchical edge bundling, which, although beautiful to look at, sometimes isn't the best way to represent networks or to bundle networks, to clean up networks. So we played around with different ways of curving lines in these diagrams and compare them to other tree visualizations. So we could use the tree to show structure, but use these arcs to show connectivity between the nodes in the trees. So both of those visualizations, like many node-linked visualizations, or many visualizations in general, kind of follow this dictum by Ben Schneiderman that you first provide an overview and then you dive into the parts you care about. But we thought it'd be interesting to kind of turn that around completely. So instead of, if there is no real way to show an overview of something so complicated, maybe don't even bother trying. Instead, let the expert who knows what they're looking for, at least somewhat, start with some keywords and just show, in this case, the proteins or protein complexes they care about. And then if they want to, they can decide which paths they're interested in, which parts of the pathway they want to see. And this is doing the same thing. There's multiple files representing different parts of a cell, or different parts of various signaling pathways. And then, interestingly, as I mentioned, some of the representations of the pathways are hierarchical, where the curator of defining the pathway has decided that it's interesting to structure the pathway in this way for whatever task they're working on, or again, they may say this one pathway points to some other file format. So we hadn't really seen anything like this. I could be wrong, but as far as I could tell, there was no existing network visualization that let you incorporate a hierarchical visualization within a node link diagram. So this project called BranchingSets, one lets you interactively branch through all the possibilities that are there, and also lets you zoom into these protein complexes to see the subcomplexes of the proteins involved. And this was presented at BioViz 2 years ago. A more recent project is time arcs. This is a collaboration with Tuan Dang, who's now at Texas Tech University, but he was at EVL for a while. Is a project called time arcs that was published in Computer Graphics Forum last year. And the idea here is thinking about some of the potential evolution of knowledge itself, or understanding of some of the conflicts in knowledge. And so since a lot of this information was gathered through reading papers, PubMed articles, we thought it'd be interesting to visualize where the knowledge came from. There are tools that let you look at a pathway and kind of roll over it, and it shows you, what paper it came from. But it's not. We hadn't seen one that was organized in a timeline fashion. So we wanted to visualize the evolution of our knowledge about different protein interaction networks or specific protein interactions, and to depict the quote-unquote popularity of proteins over time, to see if particular proteins tended to be studied together in different experiments. And the project indicates whether or not our knowledge about the functionality of proteins is in conflict, which is a potentially very interesting area to explore. If experiment one, which is supposed to be peer-reviewed empirical paper, says X, and another paper that comes out 2 years later says the opposite, that could be a good point to start a more in-depth investigation to see why there's conflicts, to see in which context is something true and which context is something not true. So in this visualization, you can see it goes from earlier in time to more recently in time, and the different colors indicate kind of a very high-level overview of what these protein complexes are doing in a pathway and how they're binding to other proteins or how they're affecting other proteins. Different colors mean they increase activity or decrease activity or bind or unbind. And one of the things that this project lets us do is to identify supporting evidence. So in this case here, we can see that PCAF was first appeared in the literature. Someone studied it in an experiment in 2002, and at least in the database we had at the time, which again is about, I think at the time was 250,000 papers, no one else wrote about that protein until 2008, at which time another paper came out and noticed that PKF had this binding relationship with 5 other proteins. And then another experiment came out a few years later, which showed it had also bounded with this MAML protein, but also something else. And then that same information was reproduced 2 years later. So it's just a starting point for experts, systems biologists or molecular biologists to continue research. In this case, maybe if they're doing research on PCAF and MAML, they have some evidence that indeed this is knowledge that has been shown to be true for the last decade and a half. On the other hand, the software also shows conflicting evidence, which perhaps is more interesting. It doesn't mean that one paper is wrong or one paper is right. It just means maybe the context changed, or maybe the experimental methods are more refined in future versions. But here you see an example where, kind of strikingly, the adds modification or moves modification indications by these color is completely reversed in a paper that was published in 2012 than one that was published in 2003. So, the feedback we got from the molecular biologists that indeed this happens a lot, and this is exactly, these conflicts are potentially starting points of where to do further research. And it's especially interesting if they're studying maybe different, let's say different cancers and different cell lines or in different organs or in different species. You might expect that something that's true, let's say in a rat, would be true in a human, but maybe it's not. And so this could give you some evidence that you shouldn't trust everything that was done in a study that was done with one species. And so this technique, just again a tangent, although it was originally developed to look at literature of biological pathways to find supporting evidence or conflicting evidence, we also applied it to various blogs about politics. So we could do some clustering, find when certain terms appeared in certain years and when they appeared together and when they didn't appear together over time. So the last project I want to touch upon is one called Dynamic Influence Networks, which is a project done in collaboration with Walter Fontana, who's a systems biologist at Harvard Medical School. So this project is a success story for the visualization class I teach. I decided last year that instead of letting students pick any data set they want, which is what I usually do, they could pick any data set they want, but they tend to pick ones that aren't, to me at least, particularly exciting. So I thought instead I would find scientists or colleagues that I knew who had interesting data sets and who wanted visualization or wanted people to experiment with different visualizations of their data and assign those to students. So I found 6 collaborators, including Walter Fontana, but also some folks who did river geomorphology or who worked at the UIC Public Library and some various other ones. And then they had to choose which data set they wanted to represent. One team wanted to represent Walter Fontana's data, systems biology data, and they came up with this nice visualization, which, even though it's very messy, at least has some kind of nice aesthetics to it. And he became very excited about it and wanted to continue the project. So we turned this into a full-fledged project after the class was over. And the project is called Dynamic Influence Networks, and it's an ongoing project that I'm excited about. And it uses the output from rule-based models or simulations of rule-based models of protein-protein interaction networks using a rule-based modeling language called Kappa and a simulation engine based on Kappa called Kappa Simulation or COSIM. Some of the BioVis people here might be familiar with Kappa, but also with a similar project or similar language called BioNetGen. They're very similar, and in fact, the visualization could apply to either one pretty easily. But as I said, there's more than 250,000 proteins in a cell, potentially up to 1 million in a human cell. And there's also 10,000 different protein types in every human cell. So even if you just want to represent the types and not the individual proteins, you'll still run into a huge problem with visualization. And each of those proteins can be in different states at different times. So the idea here is rather than visualize individual proteins or protein types, instead we visualize the rules about how they are likely to function given the presence of other proteins. And more specifically, Walter Fontana wanted to visualize the influence that the rules have on each other and how this influence changes over time. And this enables researchers to analyze the dynamics of the mechanisms of the interactome, or specifically of the protein-protein interaction network, and to reason about why these mechanisms give rise to coherent systems behavior. So after some iterations, both on our own and both with some feedback from systems biologists, we came up with I think a nicer visualization that shows the rules themselves and the influence the rules have on each other as we scrub through time, as we make this kind of movie, this dynamic movie of, or what's sometimes called a network movie of the state of the system, how often the rules fire, so in this case here, let me try and break it down quickly. So the nodes themselves indicate rules and how often the rules are fired in the simulation in a given time period. So the simulations can, they can run for as long as they want. Usually they're interested in seeing some type of behavior in a coherent system. If it's not, if it doesn't blow up or completely dissipate, if it's a coherent system, usually there's kind of oscillations in the system. So you don't need to see it for all time. You can just see it for a portion of time. So the simulations usually try and find a starting point, see how it changes, then how it kind of comes back to the original layout, original state. So in this case, again, the nodes are how often these rules fire. The links indicate if a particular rule is influencing the activation of another rule, either positively or negatively. And because this alone can be kind of messy, we're using this automated clustering technique to group rules that tend to fire together at a given time. And then on the sidebar, you can click on any particular rule to see a quick overview of all of the influences it has, positive or negative, on its neighbors. And we also have what they're calling the observable chart, or the phenotype chart, a visualization of other information that's not necessarily represented in this particular network, but it could be interesting. So this is an overview of the populations of certain proteins that they care about. So those populations aren't represented in the diagram in the view here, but the rules that govern those populations are presented. And so this gives you a sense of, it's a little bit hard to tell, but over the course of a few time steps, you can have dramatic changes in the rules. And again, in this case, we're clustering per frame, so these clusters kind of appear and disappear. And in all four views here, we have the same rule highlighted, and you can see that not only does the rule stop affecting rules at some points, but it also actually switches whether it's a positive influence on the rules that it does stay connected to. So depending on how various thresholds are set up, you can still get to these states where the network visualization is kind of messy. So we allow users to do their own clustering if they want to override the default clustering, and to spatially pin certain. So we're using a four-structured layout where nodes are trying to stay away from each other. Nodes that are highly correlated tend to be close to each other, but it may be a user wants to see what these rules are doing, even if they're not firing together, so they're not clustered together. They're interested in, maybe do they have some interesting temporal pattern they want to keep track of, so they can group them in a single place. They can color them as they want to in order to more easily identify them, and they can, if they're interested, they can take some time to kind of augment the automated layout to make it a little bit easier to see. Go ahead. Yes. So, so to resolve that problem, I mean, it may be in some cases that's exactly what you want to see. You want to see this threshold change, this kind of punctuated equilibrium where all of a sudden you have something that looks totally different. But in other cases, that's distracting. It kind of it distracts. It introduces flickering or a temporal incoherence. So I have modes. I don't think I have an image of it, but you can even see in this clustering menu we have three options. One is frame per time step or per frame. One is global, so you actually do clustering on the entire simulation. And one, if you want to kind of have it be more smooth, you can set how many frames ahead and behind you're using to cluster. And that kind of can mitigate some of the flickering if that's what you're interested in. But in some cases, you actually do want to see that flickering. It's actually what you're looking for. So we give the user the option to go through both of those. And in certain cases, again, even though the rules are a pretty dramatic summary of all of the protein types and, of course, of all of the proteins themselves, it can still get kind of dense. No-leak diagrams get dense even with around 50 interconnections, especially in situations like this. So we have another visibility threshold slider where you can tune it up or tune it down so that only rules that have an influence above a certain threshold will be visualized. And here's a clear example where as we increase the threshold, once we get past a certain point, you only see these, but you no longer see these ones. And so potentially you can see some of the data that might be obscured in the visualization. So this is ongoing work, but I think it's kind of interesting in general to think about a network visualization, not in terms of visualizing the network, but in visualizing the relevant rules that govern the behavior of the network. And this is especially interesting maybe for dynamic networks. So I think there's a lot more work that needs to be done in it, but it's kind of an exciting start. And working with Walter Fontana, who helped develop the COPPA language, it's been a great collaboration. We have a paper that's in submission that uses these dynamic influence networks to investigate a rule-based simulation of circadian oscillatory cycle of cyanobacteria involving the Chi-C protein complexes. And it's always interesting being a visualization person, as you know, because you get a chance to have conversations with all sorts of people. I'm not a systems biologist, but I had a chance to learn about some of these circadian rhythms. And they've actually been able to extract these 3 proteins and put them in vitro in a test tube and actually watch these 3 proteins mimic the circadian rhythm. All living things, sleep at night and wake up in the day, sometimes at different rates. But they found that these 3 proteins alone in various complicated states, even though it's 3 proteins, it's actually pretty complicated, are enough to show evidence of this universal circadian oscillatory process. So the paper's about showing that this rule-based modeling can actually mimic that, and the next steps are to apply this approach with systems that are unknown, more complicated systems where people don't actually know what they're doing, what the systems are doing, and see if this rule-based approach in combination with empirical wet lab approaches can give rise to some interesting scientific progress. And then ongoing work is we do have this nice visualization, I think, of visualizing the rules, but being able to kind of move between that and between different visualizations where you actually do see the protein types or proteins themselves would be interesting, but it's kind of complicated to do. So I just want to end and take questions, but just say that some current research areas I'm working on are this idea of translational visualization, where you incorporate context, storytelling, maybe language, and simulation to understand systems, and not just the data itself, but the systems the data is part of, and to use visualization to illustrate the relationship of data to meaning, action, maybe policy, and maybe personal experience. And then secondly, although I didn't talk about it too much in this talk, I've been very interested in this idea of immersive analytics using augmented reality, virtual reality, computer graphics, mixed reality, different techniques to make visualization richer in a meaningful way to support and enhance data exploration and analysis. And I think that's all I want to say. So thank you very much.